All right, good afternoon. That was fantastic. So the students, I really enjoyed that. So really well done. They organized that. They wanted to do it. They organized it. They put it together themselves. That was very cool. Okay, so I'm just going to read this bio sketch for Dr. Gros because he has done so many things, and it's just much easier for me to go straight forward through. So I'm very pleased to introduce Dr. Gros. He is a scientist at the Robarts Research Institute and an associate professor in physiology and pharmacology at Western University. He's also director of the CFI-funded Laboratory for Cardiovascular and Metabolic Phenotyping. He's chair of the Scientific Program Committee for Canadian Hypertension Congress, and anyone interested in hypertension research, you really want to touch base with Dr. Gros. Dr. Gros was a recipient of the Evelyn McGloin Fellowship Award, New Investigator Awards from the Heart Stroke Foundation of Ontario when he first started as a faculty person, and a Young Investigator Award from the Canadian Hypertension Society. The focus of his research program is cellular and molecular mechanisms involved in the regulation of vascular and cardiac function and cancer. So basically he's got cancer, he's got cardiovascular, he's got the heart, he's got the vessels, it's going in lots of different cool ways with it. Dr. Gross's lab focuses on the role and regulation of G protein coupled receptor signaling pathways, as well as both the role of the neuronal and non-neuronal cholinergic system and their contribution under physiologic and pathologic conditions such as hypertension, heart failure, and cancer. I'm really glad I teach physiology. To get through all these words is amazing. The Gros lab uses a range of biochemical, cellular, and molecular techniques, as well as integrative approaches in genetically modified mice and human subjects to gain a better understanding of the development and progression of these various diseases. So, Rob, Dr. Gros, please accept this plaque in honor of your visit. Here is a distinguished scientist to the Center for Cardiovascular Investigations.
Perfect, thank you very much.
And the title of Dr. Gross's talk is The Female Sex, a Newly Appreciated Risk Factor for Cardiovascular Disease, a Role for the G Protein Estrogen Receptor.
Perfect. Thank you very much. Can you hear me, the camera guy? Because, you know, got to make sure it's good. First of all, thank you to the organizers for inviting me. It's lovely to come back here. I think I was here a couple of years ago for thesis defense. As Tami said, my lab does a lot of different things, and I've just picked one of them today. I think it kind of fits in with the theme that's going on of some of the posters that I saw today. And it's just a story that we've been working on, trying to understand how women's health maybe changes, but more importantly, how an estrogen receptor may actually play an important role. So what do we know about women and cardiovascular disease? If you ask most people, they say, hey, why worry about cardiovascular disease in women? Women always seem to live longer than men. True. Women are less likely to die from cardiovascular disease than men. And women are less likely to have hypertension. So why are we worrying about it? So what is the big deal? Well, it turns out that actually probably that's not the case. If you go to the literature and look at it very carefully, women are catching up, and if not outpacing men. And that all sort of started probably 30, 40, maybe even 50 years ago, when at that time, cigarette companies started to sort of cater towards women because women wanted to be more like men. And so Virginia Slims is one of them. The reason Virginia, woman's name Slims, because they were much thinner. And sort of they have that slogan where they took these old pictures and sort of said, you've come a long way, baby. The problem was that all these ads were sort of pushing, because if you show a doctor smoking, it's okay for women to smoke. And they actually pushed it so far that they actually started pushing cigarette smoking as a good thing for pregnant women. And this is Jackie Kennedy from JFK, who also was caught smoking while she was pregnant. And it was the norm. So the whole idea was that if your dentist told you you could smoke, your doctor told you to smoke, well, why not geeky scientists too? You know, why not them too? But the interesting thing is that that was the whole push, because they really felt at that time that women wanted to be more like men. So women should also then smoke. And this came from a reputable scientific organization. The reason that I say this is because if you, as I said earlier, go to the literature and start actually carefully dissecting out what cardiovascular risk is in women, actually it is pretty substantial, and it's very similar. So what is the sex gap in cardiovascular risk management in women? Women after menopause are more likely to have cardiovascular risk factors compared to men. Women with cardiovascular risk factors are less likely to be treated appropriately. That's also true. Women with atherosclerosis are less likely to be diagnosed with coronary artery disease. Women with atherosclerosis are less likely to be treated for coronary artery disease. And women treated surgically for coronary disease are more likely to have complications and death. And the biological basis for all this is really still a big black box. So have we really come such a long way, to use that slogan from those ads? And it probably comes back from what you all learned in biochemistry, certainly about this, and now of course we have added gender, but really trying to understand what sex hormones play and what role do they play in the progression from when we're born, when we turn an adult, and when eventually we become a diseased patient. And sort of for our lab, it's trying to understand what does the estrogen receptor, or this newly discovered estrogen receptor, this G protein estrogen receptor, actually play a role in this progression. So here I'll go through some of the stats. So really, cardiovascular death is an equal opportunity disease. Yes, there are those, and you saw earlier, that there are those diseases that are more likely to kill men. But if you looked at for heart failure, it's probably more likely to women. If you look at stroke, probably more likely to women. So overall, cardiovascular disease is very similar between men and women, despite what you think. Now you got to remember that a lot of this is because it's post-menopausal, and the post-menopausal woman starts to look a lot more like a man. The same is true right here. If you look here at mortality rate in the hundreds of MIs, women are more likely to die from when they have an MI. Here, less likely to be treated effectively. So if you look across between men and women, it's probably more likely that women are less effectively treated for the diseases. The reasons for that are unknown right now, but it may be because they just complain less when they have disease as compared to men. Of course, even bypass surgeries, women are always less likely to get bypass surgery. Even though they are clinically needed to get the bypass surgery done, they're probably most likely to get those surgeries. And even when they do, they're probably more likely to have mortality rates that are higher than men when they have had the revascularization surgery. So all these things sort of kind of query about why women are actually catching up, and if not so, surpassed men. So what have I said so far? That coronary artery disease is an equal opportunity disease, sexually speaking, of course, between men and women. Women catch up, especially postmenopausally. Women with established atherosclerosis are probably less likely to be diagnosed properly, more likely to die from their MI, probably less effectively treated for whatever disease they have, less likely to be treated surgically, and more likely to have even complications or death following revascularization. So what is this risk profile of postmenopausal women? Because it is worse than men. They tend to have higher BP, they probably have higher LDL levels, and they're more likely to have multiple risk factors. So these are all these things that have been shown out in multiple clinical trials and studies. So what are the determinants of sex-specific cardiovascular risk in women? Well, we already sort of mentioned some, culturally and socially and behavioral, even because some of them are pushed. The other thing to remember is that a lot more women over the last 40 years have sort of entered the workforce. So not alone doing the homework, they also have now the added benefit of working like everybody else, but the added stress. But of course, many biological factors. If you look at it biologically between men and women, women are at higher risk for to form thrombosis, more likely to have endothelial dysfunction. The atherosclerotic plaques that they do form in women are less fibrotic, so they're probably more likely to rupture. And there's more microvascular disease. And the cellular and genetic disk, beyond having the Y chromosome, and the role of estrogen is unclear, especially postmenopausally. As I showed you this earlier, right, we've been interested in trying to understand what the sex hormones play, and especially estrogen, in women's health. And sort of was mentioned earlier in one of the posters that both the Women's Health Initiative as well as the HER studies both reported no cardiovascular protection benefit in the treatment of estrogens in postmenopausal women. If anything, there was an increased risk, and that may be in part reflected what you saw in those data sets. So why is there a difference between women when they're younger and have lots of estrogen floating around, and probably the right receptor combinations floating around, as opposed to when they've established atherosclerosis when they get older and more likely to be postmenopausal? And there's of course many different factors that you've heard about today that play a role in either the contractility of the blood vessel but also the plaque forming. And it's possible that the receptor populations or the combination of receptor populations have altered in the older women in such a way that they're actually now at higher risk. So what do we know about estrogen in the cardiovascular system? Well, we know that estrogens are important, both physiological and pathophysiological regulators of cardiovascular function. And we know that traditionally these cardiovasculars have usually focused on the classical steroid receptor, so the estrogen receptors or the ERs as they're known. And so this is what you probably all remember from your biochemistry text, that estrogen can bind to the receptor does what it's supposed to do. And a lot of this activity is very long-term. It takes time for these receptors to do what they do. It turns out that probably more recently, some of the effect of estrogen in the cardiovascular are probably more mediated by a new protein that was discovered about 10, 15 years ago. It's the G protein estrogen receptor, GPER, also known as GPR30 because it was an orphan receptor. And there's now increasing appreciation for the actions of estradiol acting through this receptor in the cardiovascular system, as opposed to this balance between the estrogen receptor old school versus the estrogen receptor new school. And as I said, this receptor was discovered in multiple labs, and sort of over the years we have tried to figure out what the role of this receptor is in cardiovascular systems as well as other systems, as Tami mentioned. So what are the characteristics of GPER? Well, it's also known as GPR30 because it was, as I said, an orphan receptor. Initially was an orphan receptor, but very quickly it was shown that it actually bound estrogen very well. And more recently, we have shown that this chicken wire estrogen can also bind aldosterone, which is another chicken wire. And it's then the reason that chicken wire can bind to different receptors. And the other thing is that it's widely expressed in sexual organs well beyond, obviously, the vascular cells, liver, hepatocytes, and kidneys. And you can sort of see in this review paper that the pleiotropic effect probably of GPR are found in multiple tissues, and that's why it's been of great interest in our lab, trying to understand what role this receptor, and of course estrogen, that men and women both have, actually plays in the protective effect or the detrimental effect of this receptor. One of the very earliest studies we did was sort of to try to understand what the difference was between the estrogen receptor proper and then the then newly discovered GPR30, or now known as GPR. And this was published in 2009, so quite a while ago when we first started the work in my lab. When we overexpress the receptor, so either GFP or the ER alpha or the GPR30, you can see there's a divergent effect between what we saw with estrogen in interacting with estrogen receptors or estrogen interacting with the GPR30. And this was done in smooth muscle cells. So clearly showing you that there's divergent activation of the pathway. Even though they both respond to estrogen, they seem to have different responses. And we sort of came up with this kind of crude schema where we thought that estrogen acting through the proper estrogen receptor had its activation pathway, and then GPR30 interacting with estrogen would activate potentially different or concomitant pathways, but the ultimate end having different effects on either ERK activation in the case or apoptosis. And then we started sort of thinking about the balance. What does this balance actually mean in the receptor system? And does that actually matter when you actually have an injury? So what we did back then was fairly easily. We did actually vascular remodeling in a rat carotid artery injury model that I learned, like Tami, in Toronto many, many years ago. And actually we started looking at what happens when you do this. Well, if you, of course, compare non-injured for the injured, you could clearly see that there is an injury model, and it was very easy to be reproducible. We then started looking at what happens when you activate the GPR or the estrogen receptor, or what happens to these estrogen receptors when their vascular model is there. Well, the first thing we observed very quickly is that actually in an injured blood vessel, GPR actually disappears. And it was both shown at Western blot as well as qPCR. So clearly showing you that this receptor following injury seems to disappear. So when the receptor is gone, it doesn't matter how much estrogen floating around, there won't be any protection. The other thing that we very quickly realized is that when we can overexpress it, we can actually reverse the damage that was done, or sort of lessen the damage that was being done. And I sort of just summarized it on the bottom here where we compared male and female rats. This is the injury and when we introduced GFP in the blood vessels, and this is when we introduced GPR in the blood vessels. So you can see that females had already a bigger injury as compared to males, but both cases were we able to, when we reversed the idea by overexpressing or reintroducing GPR, we can actually reverse the amount of damage being done. So clearly showing you that estrogen and interacting with these receptors had a protective effect. Of course, we went then on to say that either increasing it or increasing the other receptor, so we could either increase GPR, we could actually lessen the, or removing the estrogen receptor proper or the ER alpha, you could have that same response. So either increasing or decreasing ER, you could actually then show that you get a similar effect. So it was all about this balance between the GPR and the estrogen receptor. And that's sort of what I sort of summarized to you earlier, but that was also then held through when we did a vascular injury. Well, how do you then try to translate what you see in a cell or in an animal to the human population? And that's always the thing that I tell students. You got to go to the literature and find out what have people done in other fields? And as Tami mentioned, I like working on cancer. We've developed many cancer models. Cancer is the field to go if you want to look for human SNPs. So these are single nucleotide polyvariants or variants that are there, because these guys, that's all they seem to do. So when you go to the literature, you type in cancer and your favorite protein, there's probably a good chance that somebody in the cancer field will have done it. But when we did that, we found that the cancer field has discovered 3 single nucleotide variants. The one that we focused on was the one that we thought was probably the most relevant, in the sense that it was the easiest to detect in our human population. And this is the P16L GPER variant, very common. The allelic frequency is 20%, so it occurs quite frequently in the human population. And then what we tried to do is overexpress this receptor initially in cells to sort of see what does that one polyvariant does to the receptor expression, which it did do nothing, but more importantly, what does it do to the function? And it actually turns out it's hypofunctional. So this is the wild type GPER. You see we have interested either looking at GI, which is this G1 specific stimulus of the GPER. So you can clearly see that wild type response here. This is the polyvariant. And it doesn't matter what assay we did, you can clearly see that these are hypofunctional. So then we thought, okay, great. So we now have identified a receptor in the human population that is hypofunctional, so it doesn't function as well, but it's expressed just fine. So then you ask the same question, how does this hypofunctional GPR genetic variant then affect the development of cardiovascular risk in a human population? So we were looking at blood pressure development as well as lipoprotein metabolism, because those are the 2 key factors that we had sort of identified would be relevant to the population. The way that we did that, actually, we just recruited a whole bunch of students willingly, as well as staff willingly. No, they all came voluntary. And then we just drew a little bit of blood and then just took the base parameters of what their blood pressure was like and whether they expressed or didn't express the variant. As I said before, we knew that this receptor is expressed in both the veins and arteries. It seems to vasodilates when activated. It regulates vascular growth, and it alters responsive vascular injury, as we had shown. So we knew probably that this receptor would probably play an important role in both the cardiovascular and the lipid metabolism in the human population. So what did we find? Well, when you first look at the population, so this is the wild type, and these in purple are the ones that are the variant. You see that we have lots of students and staff in our population. When you looked at mean arterial pressure, now remember that these are otherwise healthy. These are students like most of you that just came and said, yes, I'll take the 20 bucks for the blood draw, right? So their mean arterial pressure was slightly elevated in this population. So this is both male and female. You can see slightly higher. Both the systolic was a little higher, diastolic was getting close, but the mean arterial for sure was higher, as was the systolic. Now this is both males and females, and then what you have to do for all studies, you actually have to separate the 2 populations. And when you do that, there's something interesting apparent. So when we looked at mean arterial pressure, systolic or diastolic pressure, when we looked in the males, although the blood pressure is higher, which we see all the time in males versus females, there was no significant difference whether you were a male with the wild type or the variant. And it didn't matter whether you looked at systolic, diastolic, or mean arterial pressure. In women, on the other hand, you had that variant, you had a slightly elevation of your blood pressure. And that held true both in the diastolic and the systolic. So it didn't matter to us which we looked at, but just having this one single mutation in this one estrogen receptor was enough to drive your blood pressure. Now, mind you, these people aren't hypertensive, they just have elevated blood pressure. Well, if you have elevated blood pressure long enough, because most of these are students, right, eventually that will develop in hypertension. But we thought it was an interesting observation to have that one polyvariant show that a slight increase in blood pressure. What was even more fascinating then is when we went to the hypertensive population. So on the 4th floor University Hospital, we have a hypertension clinic. So you just go and get blood samples there. So when we did that, if you're a male, it doesn't matter whether you're wild type or the variant, that ratio of the 60-40, so that's the 60, right, 60-40 split that you see between the wild type and the variant is because it's a 20% allelic frequency. It doesn't change whether you look at normotensive or hypertensives. In the female population, on the other hand, there's a high proportion of the variant in those women that have hypertension. So that goes to show you that eventually these young people that have slightly elevated blood pressure eventually develop into hypertensive patients, because these are what we call hard to treat hypertension. They come to a tertiary care center to be looked after as opposed to a family doc. So we knew there was an effect on blood pressure, and we usually know, and as many of you know, that blood pressure and dyslipidemia or dyslipidemia, blood pressure, hypertension, they all sort of come together. So we thought, fine. We knew that estrogen regulates LDL metabolism. That had been previously shown before, that low density lipoprotein or LDL cholesterol levels increase postmenopausally, had been shown. People had shown that estradiol increases hepatic LDL receptor levels, so good, and that estradiol decreases PCSK9 levels, which is sort of that anti-thing that seems to degrade LDL receptor. But we did not know what the effect of ER versus GPR was in our population. As I said before, that we knew that these things are expressed in many things. We know that GPR is expressed in epidocytes and hepatocytes. We knew that GPR knockout mice yield the higher total cholesterol, triglycerides, and LDL. So we knew there may be some effect within the population. Now the first thing that we did is, and that's the nice thing sometimes to being in a place where you have somebody that is a geneticist and has a large patient population. We actually looked in the Rob Hakeley population at the Hutterite. So this is samples that he's collected over many, many years. And this is sort of like this inbred population. So you kind of have this ability to sort of have all these family members. When we looked in this whole population, so there's 415 people, when we looked at the wild type, so this is the full homozygous wild type, or the heterozygous, or the homozygous that has the mutant. So this is the mutant, this is one copy of the mutant, this is having both copies of the mutant. When you look in this population, both total cholesterol and LDL cholesterol goes higher in the population that are homozygous for this mutation. You can then split it up between males and the females are the one that carry the entire population. So the females are the ones that when they have this mutation have higher cholesterol levels or LDL levels. And then, of course, what you have to do in most of these trials, you actually have to then get some of your patients or your subjects back in our trials, and we call that the London cohort. So now we recruited 339 females in this case and did the same thing. So this is the homozygous wild type, the heterozygous, and then the homozygous mutant. And if you look here in LDL, you can see this is in the normal population. When you have one bad copy, it goes up slightly. When you have two bad copies, it goes up significantly. So clearly showing you that having this single mutation in this female population, and again, not in the males, there's an effect on your LDL levels. So does GPR then regulate LDL metabolism? Because the hypofunctional variant, the P16L, is associated with higher levels in female subjects. So our hypothesis was this GPR activation increases LDL receptor expression. So this we did then in isolated human hepatocyte, and this is one of those other important things to take home when you're a student. There's lots of animal models. So there's mice, there's rats, there's humans. Those are all good models. When we initially went to look at mouse models or rat models, those animals do not express that same receptor that we see in the human population in their livers. They have it everywhere else, they just don't have it in the liver. So that would make it a really poor model. So then we were forced to actually then get isolated human hepatocytes because we wanted to repeat the experiment. And what we did there is we stimulated the human hepatocytes with this analog, the synthetic analog of estradiol or G1, which is a specific agonist for the GPR. What we showed there is that when we increased the dose of G1, we can actually increase LDL receptor expression in these human hepatocytes. That makes sense. So if you have a functional receptor and you stimulate it with estrogen or with G1, LDL receptors go up. So they control your cholesterol levels. When you have a mutant receptor, it doesn't matter how much estradiol you're floating around, you can't increase the LDL or the LDL receptor. So needless to say, you end up with more LDL in the circulation. Then we went to sort of show the belt and suspenders approach, where you sort of use agonist and antagonist to sort of show what the expression is. So again, this is the agonist. Of course, if you add the antagonist and the agonist together, you can see that you have a major effect, that you do not have overexpression. Atorvastatin, which most of you have heard about, is one way to get rid of LDL by increasing LDL receptor expression. Atorvastatin and the antagonist to the GPR don't do anything, and G15 on itself doesn't do anything. So this is doing it one approach, using an antagonist. And then the suspender approach is where you actually do shRNA, so you decrease the receptor expression. So this is in a wild type cell with G1. You can see you have lots of activation. When you knock down the receptor and you add G1, obviously you have reduced the amount of receptors there. And of course, when you act the agonist, you can't stimulate too much of the LDL receptor. Similar if you did with atorvastatin, it wasn't affected by the siRNA. So clearly showing you that the GPR plays an important role in mediating LDL receptor expression and hence LDL receptor, or LDL levels. So what are the conclusions here? Women have caught up to men in regards to cardiovascular risk, especially post-menopausally. Women are more likely to suffer complications following vascular injury. And among the multiple determinants, the balance between the GPR and the ER may be really important in trying to understand what this receptor does in a patient population, as I showed you before, that women that have hypertension are more likely to have this dysfunctional receptor and are more likely to have higher LDL blood levels. And again, the same kind of idea, of course, right? Someday a woman for president. That was also from the Virginia Slim, and they used that same slogan, "You've come a long way, baby." But I think women have unfortunately caught up to men, especially postmenopausally. But I think there may be potential ways for us to come around to try to understand what these receptors, which occurs in about 40% of the population, may do in cardiovascular disease in women. So I'd like to acknowledge the technicians and the students that helped out, and my collaborators from different places, of course. Ross Feldman never seems to stay long. He's at UW, was at Memorial, and is now in the University of Manitoba. Rob Hagley, the geneticist and endocrinologist, and Jeff Pickering, the cardiologist, and the funding sources, and I thank you for your attention.
All right, so we'll now take questions for Dr. Gros.
Peter Bax, he has a question,
Yeah.
guaranteed. I didn't even have to look.
All right.
Since this is all about me.
It's always about you. It always was. When I was in Toronto, it was about you, right, Tami? It's always about Peter.
So you never really, so the one question I have is that, I have lots actually, but you know, I mean, okay, so you have these receptors that can signal alternatively from the nuclear receptor. Yeah, so,
yeah.
right. Well, I mean, what are the numbers? Are there gender differences in terms of just the numbers? So you showed that the males really don't have any difference in terms of polymorphism, but do they express way less receptor?
They do what, so from the preliminary data we have, the limited data we have, because it's much harder to get tissue samples from people, right? I mean, the liver samples is a little easier because to get the sac, but to get different tissues, to actually get the level, the expression levels, it's much harder to do. When we look at the hepatocytes, which we have had for males and females, both they're usually in their 50s. Expression patterns looks very similar, but expression levels look very different. That's based on protein. What we think it is, it's probably the amount of estrogen or estradiol that we've added there as well, or other hormones. So I showed you aldosterone works on this receptor as well. Now, males, females, both have very similar levels of aldosterone that fluctuate. But I think it's a combination between having the GPER and how much estrogen receptor property of the traditional receptor. And it's this balance that maybe offset more in women so they become more sensitive to. We have noticed that it's as women age that the amount of this receptor expressed with a wild type GPER decreases. But we've also seen this in injury. We've also seen this in hypertension. So as women become hypertensive, the amount of this receptor seems to be going down as well. So I think it's a very fluid kind of receptor. I think it's more of the balance, because I don't know how much of the estrogen receptor has changed in women as they age or if they have. I think it's more of this balance. So because they have divergent effects on some of these systems, that balance may actually be more important.
I have a question about the potential for cyclic fluctuation in expression. So there's some data to suggest that endothelial function is sort of better when women are in the high estrogen phase of the menstrual cycle, and that even angina is less in the high estrogen phase compared to the low estrogen phase. And then there's some evidence that estrogen receptor alpha expression actually changes cyclically across the menstrual cycle, so it's higher when estrogen is high. Is there any evidence of that for the
Interesting, and it's a good question. I mean, we look, so, I mean, obviously when you get the student population, we take a whole questionnaire. We tell them where they are in their estrogen cycle. We look at whether on birth control or not. And even when we've looked at all those factors, for the GPER, it looks like it's fairly steady. I don't know what that means yet, but in the sense that when they were on or off birth control, or when they were at the first beginning or the end of the cycle, didn't seem to matter much for the expression level that we could see on the GPER. Now, was that the main focus of our initial screening? Because we didn't do it on all 39 women. We do see a difference between younger and older. We had obviously predominantly young population, so 19 to 22 years old. And then we had some staff members that were in their late 40s or 50s. We do see a slight difference there in the expression pattern. So it does look like with age it seems to go down, but we couldn't, from the base that we had, and remember this is just a questionnaire, we didn't measure anything. So, you know, and whether they took or didn't take their pills, we don't know. But as long as we looked at the broad spectrum of the young population that were on and off birth control, we could not tell the difference in the amount of expression of this receptor.
Kind of a flip side question.
Oh, sorry, yeah.
Yeah. You know, 40% of the population has polymorphism. So what is the benefit of having this type of mutation? Do you have any speculations why it might be so prevalent?
It's a good question. It may in part have to do that maybe it's about the balance. So you may have this mutation, and when you're young, your estrogen receptor, probably ER alpha, ER beta, maybe keeping this other receptor in balance. Right? It's all about this balance that we think it is. As you age or become post-menopausal, maybe that receptor, even though it's hypofunctional, starts to lose it because the driver of it, the estrogen, seems to also be disappearing. And maybe then that balance becomes more, either because estrogen receptor proper may just be happy with whatever dose of estrogen you have around. This receptor, when it's already hypofunctional, may actually become very sensitive to the lower dose, and there may be a critical cut on that. And what we haven't tried yet in animals is trying to replenish the estrogen, or estrogen in the case, and see what happens to the outcome of the carotid arteries. Because that's the follow-up study that we're doing right now in young, middle-aged, and really old female rats, and sort of see what happens. If you replenish them, we know that from the human population, you're probably going to put at higher risk. If when we're looking at just this snippet of artery, maybe we can see that when you push that receptor that's hypofunctional, when you push it a little more, with a little more estrogen, maybe there'll be some benefit. And maybe that's what came in the human population, because if it is that prevalent, right, when you push the population by putting estrogen back in, maybe those women that were more sensitive because they have this dysfunctional receptor, maybe they're the ones that are causing more of the cardiac events.
Any more questions? No? Okay, well, thank you very much, Dr. Gros.